Ladies and gentlemen, good morning. It's a great pleasure to welcome Dr. Harold Varmus, who is an extremely with it sort of person, a real Renaissance man. He is not only a Nobel laureate in medicine for his work on cancer genetics, but he lives the lives of art and science and culture sort of seamlessly. He's a musician. He writes the book that we will discuss. He is a sports person, and he delves into a lot of very, very interesting things, as you will discover, no doubt, in his own talk, and some of the discussions that we'll have later. So welcome, Dr. Varmus.
So I'm very grateful to Suman Sahai for sharing the podium with me today. Our plan's the following. I'm going to make a few remarks for about 15 minutes or so. Then I'm going to have some questions addressed to me by Dr. Sahai. And then we're going to open the floor for some questions from some of you. I am a bit of an anomaly at the Jaipur Literary Festival for at least 3 reasons. First, I identify myself as a scientist rather than a writer, and I haven't seen too many people doing that yet, but I've been grateful for the few that have appeared at the podium. Secondly, I am here in India not to come to the Jaipur Literary Festival, but to work on behalf of the National Cancer Institute, which I now direct, to try to develop collaborations with colleagues who do cancer research and take care of cancer patients throughout India. But I'm very happy that fortunately the festival occurred at the same time that I was invited to come and make a presentation and to hear many other distinguished people make presentations at the festival. Nevertheless, despite this disclaimer that I don't self-identify as a writer, I must be a writer because I have written a book, and the book is present in this place and can be signed. But I'd also note, and this may be pertinent to something that I will say in a few moments, my book will soon be available for free in digital form on my website at the National Cancer Institute, thanks to the generous and foresighted actions of my publisher, W.W. Norton. And I would urge others who don't identify as writers or write for a living to consider putting their books for free in the public domain once they feel that that's a useful thing to do. So, I'd like to talk briefly about a couple of things. One, very briefly, why did I, a non-writer, write a book? And what did I want to say? So I wrote the book because I had been invited to give a series of lectures at the New York Public Library, something that is a fairly common event in my life, to be asked to give a lecture. But these lectures were named the Norton Lectures in honor of the donor of the lectures, namely the Norton Publishing Company. And I failed to read the fine print, which said that the lecturer will submit a manuscript within a year after giving the lectures. And that the acceptance of the fee for the lectures was contingent upon producing the book. Well, after I gave the lectures, someone pointed out the fine print, and 4 years later, I did come up with a manuscript. The manuscript was not the lectures, it was something much longer, and I think somewhat better, because I thought over the course of the years between the lectures and the book, about what I wanted to say to people about my life in science. There were at least 4 things. First, I wanted to talk about how I became a scientist, because I did so in a rather unusual way. Second, I wanted to say something about what I did in science, not so much to describe the science itself, which is in scientific journals, but to say what it's like to be a scientist, what it's like to make a discovery, how one relates to one's colleagues in science. Thirdly, I wanted to talk about something rather unusual that had happened in my life as a scientist, namely how the award of a prize suddenly made me eligible to do other kinds of things. In my case, run a very large institution, the National Institutes of Health, despite no previous training in administration or running budgets or doing anything that I was then required to do. And fourthly, I wanted to talk about what it means to be in charge of a scientific institution, especially one like the National Institutes of Health, arguably the largest and most significant scientific research organization in the world, and how that affects a number of important policies that influences the lives of all of us. So I'm going to talk briefly about what I had to say on these four topics, and then we'll start to entertain questions, first from Suman and then from many of you in the audience. So I began life as the child of two professionals who were the children of immigrants. One of the remarkable things about the American experience, whether one's coming from Eastern Europe, as my grandparents did, or from India, as many of my colleagues in science in America did, is how quick the transformation is from being a poor and sometimes discriminated against person to becoming the parents of children who receive Ivy League educations and become professionals. I grew up in middle-class circumstances, went to a public high school, was not one of those children who spend all their time with chemistry sets. I enjoyed reading novels and going to the beach and playing tennis. I went off to a very good college called Amherst College in Massachusetts. The expectation was that as a young, smart Jewish kid whose father was a doctor, I too would be a doctor, but I very quickly became entranced by other things, majored in English literature, wrote a thesis about Charles Dickens, learned about India by reading E.M. Forster and watching the movies of Satyajit Ray, and then went not to medical school after college, but to graduate work in English at Harvard, where I learned Anglo-Saxon and read Beowulf and studied Chaucer and Shakespeare with Harry Levin and many other things that might have been next steps in a career toward a life in literature. But at some point along the line, I began to feel that one side of my brain was going to be neglected. I still cared about science. I wanted to be somehow relevant to the changes that were going on in society. And I decided to go to medical school after all, perhaps to blend my interest in literature with my interest in the brain and become a psychiatrist. That proved to be very difficult to do for someone who had a hard time spending an hour in the same room with a psychotic person. But I then quickly found that internal medicine was of interest to me, in part because people had stories and mysteries to figure out. Why did a certain set of symptoms add up to a certain kind of disease? And I was learning at medical school in the 1960s that our understanding of how biology works, how the human organism is organized, would allow us to make rational decisions, not mythic and superstitious decisions about how we could improve the care of patients who have diseases that result from changes in their genes, changes in their molecules, changes in their biochemical attributes. So having finished medical school, I had to make some choices about what to do next. I was living in a very difficult political era. The U.S. was engaged in a war that I did not support, and I had only a few options in life since all doctors were subject to the draft. I could either go to jail, not appealing. I could go to Canada, only marginally appealing. Or I could serve in Vietnam, very unappealing. Or I could go into the public health service, a part of the U.S. government's effort to provide services, and learn science at the National Institutes of Health. And I was fortunate, despite almost no experience in a laboratory, to go there. At the NIH, where frankly I was selected because my mentor was married to a poet who wanted to have someone in her husband's laboratory who was able to talk about poetry with her. I was then taught about doing science, not as I had hoped, by working on the biochemistry of the thyroid gland, which related to thyroid diseases that I knew something about, but instead because my mentor at the NIH was had just become interested in gene control in bacteria. I was suddenly plunged into an area of molecular biology and genetics that was just emerging as a new field. But I found that I loved it, that discovery was exciting, that being in control of experiments every day and making new observations was a thrill to me at a level that I had not expected. So at that point, I said to myself, well, I need to have some kind of relation to medicine, and I was able to learn a little bit about cancer from courses being taught on the NIH campus, and I realized that although this was an era long before we could study human genomes in the rapid way we do now, before DNA sequence or recombinant DNA technology or any of the common methods were available, that it was possible to make an approach to cancer because there were viruses in animals, viruses carrying very small numbers of genes that were able to change the behavior of complex cells, cells from birds, cells from mammals, into cancer cells. And that seemed to me an approach to understanding the fundamental nature of cancer that was approachable with some of the techniques I had learned. So I made another step in life. I was now reaching 30 years of age, an age at which people, at least in those days, were expected to have a profession. I decided to go to work in California with a man who became, for many years, my scientific partner, Michael Bishop, and we chose to work on a virus that had been discovered in 1910 by examining some features of a cancer that had appeared in a chicken that was grown on a poultry farm in upstate New York. And that tumor had within it a virus that was able to cause the conversion of a normal chicken cell into a cancer cell. But it had very small numbers of genes. And here begins the second part of the story, that is, how is science done? What did I do as a scientist? Because over the course of the next 10 or 15 years, Michael Bishop and I and our postdoctoral fellows and many colleagues around California who joined together to form a community called the West Coast Tumor Virus Cooperative, learned many of the features about two questions that drove my life for about 15 years. The first was, how do these viruses grow? Now, these viruses are actually turn out to be members of a group called retroviruses, a group of viruses that all of you in the audience are familiar with because it was learned 20 years later that those viruses include HIV, the cause of AIDS, a disease that at that point had not been discovered. The second thing we were interested in is how do these viruses cause cancer? And we know many technical aspects of how that happens. I'm only going to mention one, and that is the discovery that many of these viruses, and there are quite a number that are seen in chickens and rats and mice and monkeys and other animals, that these viruses have the ability, because of the way in which they grow, to capture genetic information from their host cells and turn that information into active machines for changing the behavior of a normal cell into a cancer cell. But that was an important observation, not just because it helped to solidify one of the many ideas that had been floating around about the nature of cancer, namely that cancer might be at heart a genetic disease, but it also provided a tool for discovering some of the specific genes that contribute to cancer. So in our own work, we showed that the virus we were working with, named for the experimentalist who first found it, Peyton Rous, the Rous sarcoma virus carried a gene called SARC that was derived from a normal cellular gene. And with the assistance of many others in the scientific community, it became clear that other retroviruses that could cause cancer had picked up other genes, all with different names that I won't recite, that exist as normal genes, important genes, highly conserved genes, genes that control the growth and death and differentiation and many other features of normal cells that could become cancerous in their actions. And indeed, a whole other cohort of investigators then showed that cancers taken from human beings frequently had mutations that changed those normal genes into cancer genes. That has become even more significant now that we have drugs and antibodies that can counter the effects of those genes and are frequently used in cancer therapy today. Now, as that story evolved, something happened in my own life to change the course of my career as an investigator and a teacher and a typical academic working, in my own case, at the University of California in San Francisco, namely that the people who make decisions about prizes decided that what Mike Bishop and I had done was important enough for us to win a Nobel Prize. That changed my life very dramatically in ways that I think turned out to be auspicious. Some people's lives are ruined by Nobel Prizes because they become arrogant globetrotters who have opinions about everything. But in my own case, while my wife would say my arrogance was already at a pretty high level, it was possible for me to find that my opinions suddenly became more valued by my colleagues, and my leadership skills, which up until then had never been manifest because I was a typical lab rat who tried to avoid any kind of administrative responsibility, suddenly I was being asked to do things because I had this medal around my neck. Well, I didn't hang it around my neck, but you know what I mean. So at that point, I was asked to speak on behalf of the scientific community if we had difficulties. For example, we had, during the course of the years just after my prize was awarded, some loss of budgetary power at the NIH, and young investigators were worried about getting grants. We had had a few cases of scientific misconduct that was undermining the public's confidence in science, and I was asked to speak to these issues. And then when we had a change in presidencies in 1992, I was asked by President Clinton to take over the NIH. Well, up until then, I had run a modest-sized laboratory. I had done some teaching. I had maybe 25 people working with me, and suddenly I was asked to run an organization that had 20,000 employees, a budget of about $11 billion, and was giving grants to thousands of laboratories around the country. So there was another learning curve in my life in the course of this prolonged adolescence, and it's one to which I found I responded reasonably well. I enjoyed doing this, and since that date in 1993, when I went to the NIH, for 20 years I've been running organizations. After the NIH, the Memorial Sloan Kettering Cancer Center, and since July of 2010, the National Cancer Institute in Bethesda. Throughout this time, of course, I've had to learn a lot about how to run an organization, raise money from Congress or raise money from donors, manage budgets, make people happy when they deserve to be happy and make them unhappy when they deserve to be unhappy. But I've also had to encounter some incredibly important issues that govern both the conduct of science and the way in which science gets done to benefit human society. And I'll mention just three of these, which were featured in the book that I put together, and still remain important features of the way science is conducted. The first was the issue of how to work with human embryo research and with stem cells. And while this issue has been, to a very significant extent, resolved, there's still some very important ethical issues on the table and some important scientific issues if we're going to make good use of our understanding of human development to use cells to both understand disease and treat disease more effectively. In the early days, when stem cells and early embryo research and human cloning were hotly discussed in Congress, there were many important debates that I was able to engage in, but I'm going to leave that for later in case there are any questions. The second issue is scientific publication. At the time that I was director of NIH, it just had become apparent that the internet and large digital databases could be used to change the way in which science is communicated. In science, writing and just telling your story and having your work read is the lifeblood of science, just as in literature, writing and publishing and having your work read is the lifeblood of artistic expression through words. And yet, scientists had not kept up with the changes in the digital world, in the internet, that would make possible, and I believe will make possible in the long run, the potential for scientists to put their work in the public domain very quickly in a way that both makes science a more democratic activity, a cheaper activity, an activity that builds on itself more equitably and advances scientific progress more rapidly. And while I was at the NIH, we were able to establish the first large digital database that can accommodate all scientific work and is gradually doing so. And we were able to show new ways to publish work so that the costs are paid and yet authors see their work available to everyone around the world immediately and for free. The so-called open access publishing movement, I later was able to advance by joining with two esteemed colleagues to form a publishing house called the Public Library of Science, which many of you will know as PLOS, P-L-O-S, which now is the largest publisher of scientific work in the world and makes science immediately accessible to everyone, whether they're a professional scientist or a teacher or a journalist or simply somebody interested in scientific development. Science not just in medicine, but science of all kinds. The third topic I introduced in the book and want to just mention here briefly and open for questions is the issue of global health, paying more attention to the discrepancies in health in different parts of the world. Indeed, it's one of the reasons why I'm here in India for these couple of weeks to try to be sure that science in the U.S. and science in India or other parts of the world are conducted in a collegial fashion and that people are sharing technologies and information in a useful way and working toward our own goals in a way that takes advantage of advances being made in different countries. Global health is, in a sense, a new concept. It's a recreation of what we used to call tropical medicine when I was at medical school. But of course, the diseases that we are sharing, that we experience in different countries, are increasingly becoming the same. And in India, which I saw as a medical student visiting here in the 1960s as a place where there was much more tuberculosis and leprosy and other infectious diseases than I was able to see in my own country, is now a country in which we have a shared pain from heart disease and diabetes and, from my own perspective, cancer, that we need to approach together to make advances. And at the NIH, I was able to foster the growth of the nation's interest in sharing our knowledge and working together with our colleagues to advance the health of other countries. In my own case at the NIH, in the 1990s, we focused heavily on malaria, a major disease in especially in Africa, but also in parts of Asia, and indeed in India. In fact, over the last 15 years, the death rates from malaria have come down significantly, and similar work, of course, has gone on with AIDS and tuberculosis, but now the shift is occurring toward many of the non-communicable chronic diseases. And I have a great deal more to say in the book, and we can perhaps say a little bit more later on as we open this for questions. So let me just wrap up here a little bit. So what I tried to do in this book was to talk about my own prolonged adolescence that finally led to a scientific career, my interest in science not just as something which produces results that benefit human beings, but also as a very human activity that, in my own case, involved a deep partnership with one scientist and the development of a strong community of colleagues and trainees who work together to make science not an isolated activity, but one that is striking for its collegiality. And then I talk about the fact that science careers can evolve, in my own case, in the direction of a career as both an administrator and an active scientist. I continue to run a laboratory even today. And finally, a reflection on the fact that science has at its core an interesting tension between the imagination of the individual who comes up with an idea to be tested and the need to show that the answer to that question has community acceptance. Science becomes real when the community accepts the ideas and recognizes the truth of the observations, their reproducibility, their applicability, their fundamental truth as facts and ideas to be built upon. And it's this tension between the scientist as an individual and the scientist as a member of a community that accepts those ideas that makes science such an exciting way to live one's life. I'd like to turn to my colleague, Dr. Sahai, for a couple of questions. I'm going to sit down next to her, and then I hope we can open the proceedings to some comments and questions from all of you. Thanks very much.
Thank you for those insights, Dr. Varmus. You've really got a huge palette from which to pick your questions. But there are parts of you that are particularly interesting. You're bridging the worlds of art and science, which you've done consistently. I mean, even when you moved into science predominantly, you continued to bridge the world with all kinds of activities. And I think your Nobel acceptance was one such homage to literature, where you quoted from Beowulf, which was unusual for somebody getting a Nobel in medicine. But I think that even otherwise, all along your life, you have dealt with literature and the arts. Dr. Varmus has also played very interesting music called Genes and Jazz with his son. Would you like to tell us a little bit about
Sure,
that?
thank you very much. Yes, well, I'm glad you mentioned the Nobel event, because that was a special occasion in which I was able to make use of my knowledge of Anglo-Saxon as I received this award in a Scandinavian country. Scandinavians having been the origins of many of the words that appear in Anglo-Saxon, which is the progenitor of my own language and your language as well, at least one of your languages, and to be in a beautiful hall, not unlike the hall in which Beowulf himself was being honored by others for having slain Grendel's mother. So
right.
It all came together in a very useful way. I am not a musician. I am a failed oboe player, a failed violinist. I'm a good listener. But my son is a very talented jazz musician and composer. You can go to jacobvarmus.com and hear him directly for free. I'm sorry, you can. He needs the money, but still you can hear him for free, or you can go and buy some of his CDs. But some years ago, Jacob and I decided that we were going to put together a show in which he played jazz he had composed with his group, while I told a story, not unlike the story I just told you, about how genes govern the the development of organisms, how our genomes are continually evolving, something that develops the proliferation of species that we all enjoy, but also gives rise to cancer, and then something about cancer. And I did this with a narration, with slides, accompanied by his music, accompanied by some very brilliant animations about how cells work and how DNA is replicated and expressed. And this show has been performed now at many museums in America, at the Smithsonian, the Guggenheim Museum, the Boston Museum of Science. I wish we could do it here, but it would be difficult to bring the musicians abroad.
Right. Let me go to your science life, and not so much the direct science as the science policy, with which you've had a lot to do. And not always does the scientific community in all parts of the world, and even in Asia, in our own country, deal so proactively with science policy as you described having done, and which opportunity you got at the NIH and then later at Sloan Kettering as well. You've dealt with different kinds of political leadership, and I think it would be interesting to know how you dealt with science policy at the highest political level. You dealt with the Clinton administration, and how does one deal, or how did you and your administration at that time deal with the religious conservatism which affected science policy very directly?
Right.
How did you deal with it? Are there lessons? What were your most interesting highlights?
Well, first of all, let me say that you can't convince everybody. And one of the precepts of democracy is that eventually, if you can convince the majority of people that science is important and worth supporting, then it gets supported. My fundamental precepts in dealing either with my colleagues in the administration or with members of Congress is to convince them in as honest a set of terms as possible that science is something that we do for public benefit, that we recognize that most science is supported, at least in America and certainly in India, by money from the government and therefore from the people, that to make progress in science, you frequently have to do work that doesn't have any obvious practical applications. When we started our own work on cancer genes, it was done in a way that many people thought was probably going to prove to be a waste of money, frankly. Why should you begin to study human cancer by looking at a virus that causes cancer in a chicken? That may seem preposterous, but one could equally say, and perhaps even more clearly understand, the unusual routes through which science works by asking, why would you want to study a virus of bacteria that is unable to infect certain kinds of bacteria? Where would that lead when, in fact, as you certainly know, and as many in the audience probably know, work of that kind led to the discovery of so-called restriction enzymes that cut DNA at very specific places in the DNA, and that led to many things, to the ability to join pieces of DNA to create the recombinant DNA industry, to lead to gene therapy, which has had some recent successes, and more importantly, it led to the development of techniques that allowed us to analyze genomes completely and understand the basic blueprint by which all organisms operate, by which organisms have evolved, and our complete understanding of how life has developed in its many different forms, how diseases arise, has been dependent on these very fundamental techniques. It's important for people to understand that such work is essential and needs to be supported by the government, because industry is always looking for much shorter-term gains. So once having said that and established a relationship with Congress, you can make some progress. I would also say that it's important to have people in Congress who are prepared to listen.
Yes.
And one of the things that we face in America right now, with many other crises, the economic crisis, concerns about terrorism, the unemployment rates in the U.S., it's sometimes difficult to get people to pay attention to science. And indeed, at the moment, science budgets at the NIH, including the National Cancer Institute and many other agencies, has been declining. We still spend a lot of money on science compared to many other countries, but in India and China and other places, while the budgets are low, they're growing, and that gives people optimism about science. Our greatest concern as policymakers is ensuring that new generations of young people, especially smart young people, are interested in what we do and able to envision a productive career in science. And the relationship that we as leaders of the scientific community have with Congress and the administration that make science policy is fundamentally linked to the ability to persuade young people that science is a productive career. Some of that faith, some of the faith that the legislators and executives in government must feel, of course, has to be linked not just to these extravagant ideas about how science evolves, produce great ideas that change our understanding of the world, but also linked to some pragmatic outcomes.
The plodding science.
Yes, improved health, improved industry, economic return, and these things can all be very well demonstrated, but they're best demonstrated by looking at history and saying, yes, the U.S. has a very strong recombinant DNA industry. It produces rewards. Human insulin is now made in large amounts. We have antibodies that would never have been made without the recombinant DNA industry that return patients with breast cancer to health. It's created an understanding of the genetic origins of cancer that produce drugs like Gleevec that take a disease like chronic myeloid leukemia that previously delivered a death sentence within 5 years and given people a normal lifespan. And we can describe those consequences of science as practical consequences that are enriching for the companies that use them, that are beneficial for people who can derive benefit from them. Then Congress sees not just the mystery and wonder and glory of science, but also the practical applications and their utility in many areas of important human activity.
Two very quick questions before we open it up to the public. One is of you as a practicing scientist, as a lab scientist. Were there moments of intuition and epiphany when you came to your major discoveries? You know, there's this big story about Watson having 2 swans and saying the DNA must be a double helix, it cannot be a triple helix. Sure. There's Kekulé who saw the benzene molecule with the snake biting its tail in a hexagon. Did something dramatic like that happen with you when you came to your major discoveries? Was there a moment of intuition?
Not exactly. I mean, I think it's a very interesting question because how scientists get ideas is often difficult to fathom. And in fact, when Mike Bishop, my scientific partner, and I talk about the things that led to the discovery of what we call proto-oncogenes, the genes in cells that were captured by viruses and proved to be important in human cancer, we have different visions of how we had the same idea, but it came in somewhat different ways. For me, it came out of pre-existing literature that had a number of slightly wrong, actually importantly wrong notions, but notions that for me were provocative and raised questions. And I've always been somebody who's fairly pragmatic. Here's a question I think we can now answer, and a question that I think is going to be important. In fact, now in my role as the head of the National Cancer Institute, I'm trying to inspire my colleagues who receive grants from the NCI to envision questions in the same way by posting on the NCI website. All of you can go look at it, a set of questions we call provocative questions, questions that have not been answered historically, but I do believe that with new techniques that have been developed, it's now possible to go after them. So that's, I think, probably as best I can do to encapsulate what you're asking. But, there was always, to us, the question of how retroviruses acquire the genes that allow them to cause cancer. In the case of the virus we've been talking about, the Ralph sarcoma virus, we knew that there was a gene in that virus. But there were other viruses in the retrovirus class that cause cancer but don't carry such a gene. And I did have a Kekulia moment. In 1978, I was pushing my younger child around in a carriage while I was on sabbatical in London, and I began to think about how those viruses might actually cause cancer. And I knew from work that we had been doing on how the viruses grew that during the course of the virus life cycle, the genes of the virus, which are present not as DNA in the virus but as RNA, were converted into DNA, and that DNA was inserted into cell chromosomes. And I thought maybe the insertion of the DNA into a chromosome would somehow affect the behavior of some gene that had the potential to be a cancer gene. And indeed, that insertional activation, the integration during the virus life cycle, could occasionally, if integration were random, occasionally send the cell off into the direction of being a cancer cell.
Right.
And that proved to be right, and what's more, led to the discovery of several new cancer genes.
So this is your aha moment.
That's my aha moment.
Now, just one last one, because I think what you've done in publishing is so extraordinarily important, and it might even be as enduring a legacy, even more enduring than your work in science, and that is open access publishing. We're all beneficiaries of that, but I think there's a question mark. How does this get funded? How will it be supported?
Actually, I think we've now proven how this would work. The basic idea is to change the business model. When we started to think about open access publishing, the world operated on a traditional subscription-based model. That is, and it's one that, in fact, very strange in its conception. Scientists did their work with government money. They wrote papers and did not receive a penny for the papers they wrote. They served as reviewers and editors for journals that published those papers. The journals then took the copyright, published the papers, charged us, those of us who wrote the papers and did the work, to buy these journals, which we had to buy.
And made pots of money.
And they made huge profits. And didn't allow anybody to see the papers unless they paid the subscription fee. And once we began to understand that, we realized the only way that we could change things was to change the business model. And the business model has been changed in the following way, that the funders with grants would allow the investigators to pay a small amount to pay for the cost of editing and publication. And once that's done, you cover those costs. The journals may make a little bit of money or even no money, but there are nonprofits after all. And suddenly the costs have been covered, and this information with the lower cost, because you don't have to print on paper, you put journals into a digital form.
Format.
And now a very large fraction of scientific publication is carried out that way. There's still, of course, the possibility of downloading yourself and printing, putting some journals in printed form, but the Public Library of Science is all an online journal. The papers cost perhaps $1,000 to produce, and it's important to remember that the cost of research, at least in the biomedical field, all fields are different, I want to emphasize that, but in the biomedical field, on average, the cost of the research, at least in the United States, is about $200,000 per article. The cost of the publication is about $1,000. The research is worthless if it's not written up and communicated to people. A very small tax placed on the research allows everyone in the world to see it. And that seems to me a good business model. Indeed, everybody else seems to have voted with their feet to say it is a good model because now PLOS one of our largest journals, is the biggest journal in the world, publishing over 30,000 articles a year, and it actually makes a small profit.
Wow. Yes.
Time for questions. And I'm glad to see there's some hands.
Can we start from the left here?
Sure. You talked about the relationship between government policy and science. One of the significant advances in science is the whole movement towards genetically modified foods. And we have a huge divide between the US and Europe, which seems to be governed by the precautionary principle, one which India has by and large adopted.
I'm having a little bit of trouble hearing you. I don't know if we can make the
Genetically
microphone working.
modified.
No, I know. I hear this. I know what the question's about. I'm just not hearing every word. Maybe you can talk a little bit louder.
Yeah, I think it has to do with the positioning of the speakers, which is for the audience at the back. Briefly, your views on the acceptance of genetically modified foods, the huge divide between the US and Europe. India seems to be more in the European mode of adopting the precautionary principle with regard to genetically modified foods. Where do you stand?
Well, my own view is that, of course, not all of the information is in hand, but I do believe that genetic modification of food can have dramatic benefits for people who are hungry, for people who don't get adequate nutrition, and that a European obsession with the sanctity of food can lead to deprivation of populations that could use the benefits of science to improve the nutrition that they desperately need. So I wish more of this activity were in the public domain, not held so tightly by industry. I think that's impeded acceptance. I do understand that there are potential threats. Many of them I think have been at least partially answered. The threats to health seem minimal if non-existent, and I think that European hegemony over these issues has deprived many in the developing world from the benefits of decreased needs for insecticides, improved returns for farmers in parts of the world where drought and insufficiencies in the soil can be overcome by genetic modulation. After all, genetic modification of food is not a new thing. We've been doing it for thousands of years by a very random, uncontrolled process of crossing different plant subspecies. Why not do this in a more systematic way, changing one or two genes at a time in a controlled fashion?
Whoever has the mic, yeah.
Sir, cancer seems to be on the rise. I have cancer seems to be on the rise. I have 3 questions. Firstly, is it really on the rise or is it just better detection? If it is on the rise, how much of it is to do with the environment? And if it is to do with the environment, how seriously is the scientific community taking this?
Okay, so is cancer on the rise? Cancer incidence in numbers has gone up in many populations. It's a very complicated question to answer because let me just say a few things about the basic principles of cancer that make, so you understand why I'm going to be tentative in my answer. First, cancer is a collection of diseases that have common mechanisms but appear in different forms or due to different risk factors in different parts of the world. Overall, yes, cancer is on the rise because people are living longer, and cancer is, for the most part, not entirely, but for the most part, a disease of aging. Second, cancers are influenced by several things. First, they're influenced by the genes that you inherit from your parents. That is significant, more significant for some cancers than for others. Secondly, it's influenced by the pure chance that's created by the way in which human beings and all other organisms operate. namely that mistakes are made during the copying of our genes and the division of genes between daughter cells. So it's inherent in the way in which life works that the genome, our collection of genes, is going to be changed, and occasionally those changes can lead to cancer. The third factor that influences cancer rates is exposure to many things. There are, of course, environmental toxins that might be a major factor, but by far the biggest factor in causing the cancers we know about is tobacco use. And tobacco use drives the incidence and mortality from cancer in virtually all populations. In the US, we have seen a decline in cancer mortality of about 1.5% every year for the last 15 years, and a very large fraction of that decline is due to the fact that we have changed people's behavior with respect to tobacco use. In India at the moment, tobacco use is widespread, not so much smoking tobacco in the form of cigarettes as we did in the US, but in the use of smokeless tobacco, particularly tobacco that's chewed, tobacco that's part of pan, but cigarettes are increasingly replacing bidis, and that represents a danger. So one of the things I've been doing on my travels here in my day job as the head of the National Cancer Institute is to meet with people who are involved in tobacco control, because one of the things that we can do to reduce cancer's rising incidence in India, and I would point out that the so-called million death study carried out by my friend Prabhat Jha, recently published, indicates that cancer is the #1 cause of identifiable mortality in India at the moment. Another example of an environmental influence is human papillomavirus, which is the sole cause of cervical cancer and also the cause of some other cancers. There are now several ways to try to control the mortality of cervical cancer, which is the leading cause of death from cancer among women in India. It's a combination of screening and vaccination. There are many issues here that I don't have time to describe because I know we've got to wind up pretty soon, but I'd be happy to discuss some of those with you afterwards.
One quick one here, and then the lady in the back.
Professor Harold, here.
Yeah, there you are.
Yeah, National Science Foundation about 3 years ago started that all funders have to give data to be put on the common space. So National Science Foundation and NIH probably 3-4 years ago started that all researchers have to give data which will put on the web publicly available. So how far this has been successful, or how far those data have been used by other people to generate more research?
Okay, well, first of all, there's a requirement for the NIH to have all of its publications of work supported by the NIH placed in a full-text digital archive called PubMed Central that anybody here can access immediately, and that's working extremely well. Second, we have a policy that our data should be available in databases. That is not uniformly successful. It's highly successful with genomic data. One of the things I should mention is that the world is now coming together to try to create an alliance for being able to not only put data online, but to share data by having interoperable databases. And that global alliance, in which India is going to play a role, is now in formation and hopefully will help with many of these issues. Not all data is available as you and I would like, but these issues are not simple, but the intention is there.
Hello. Yeah. You mentioned that the life and blood of a scientist is publishing his work, making it accessible to the world at large, and have global context and applicability. Despite the popularity of open access journals like PLOS and Frontiers in the recent past, there still exists this publishing clique which is largely governed by the Western world, consists of high impact factors in journals like Science and Nature. However, catering to their standards is most often not only difficult but also meaningless for countries like India and for scientists from countries like India. So what do you have to say to that?
Okay, well, this is a difficult issue. One of the things that I didn't have time to talk about that is related to your question is the product of reduced budgets for science in America and the intense competition that currently exists among young people for postdoctoral positions, for jobs, for grants. And we have put into the hands of the major publishers like Nature and Science too much authority, too much determinative power in deciding who succeeds in science. And one of the things that we have to do as individual scientists is to say we object to that, and that we are going to publish in open access journals and in journals that don't have the extraordinary impact factor of Nature and Science, and moreover that we're going to evaluate each other not by the number of publications we have in certain journals, but by reading the papers and understanding their significance. We have been discussing at the NIH, and many other organizations have taken similar steps, the idea of not listing our publications on our curriculum vitae, but instead describing our major contributions to science, saying in plain language what we've done, what contributions we've made to discoveries, and trying to diminish the overriding effect of impact factors, quantitative, in my view, generally false assessments of work based on these numerical factors that relieve scientists of the responsibility of actually evaluating each other's work in a highly sympathetic and constructive fashion. So I think we're all going to have to work as a community to overcome both the competitive atmosphere that's been created and the unfortunate response to it that allows the editors of certain journals to make decisions about people's career that, in my view, is frequently unfair.
On that note of science democracy, we will unfortunately have to close this session, but you can swamp Dr. Varmus the moment he leaves the stage. But let's give him a big hand.